So our next speaker is Associate Professor Jonathan Keat. He is joining us from City of Hope, I believe, and he's joining us from Zoom land. So Dr. Keat is assistant professor, and he got his PhD from the University of Alberta. His main focus is in multiple myeloma, obviously, and other hematological malignancy and immunodeficiency syndromes. He has done multiple work involving the NF-kappa pathway in multiple myeloma, and trying to discover why some patients are more sensitive to proteasome inhibitors. He's also involved with the Multiple Myeloma Genomic Initiative, funded by the Multiple Myeloma Research Foundation. And his lab focuses on the most frequent mutated genes identified in patients with multiple myeloma, and his work looks at the genetic characteristic of multiple myeloma with a focus on integrating different data sets to develop better understanding of the disease. And this morning, he will be doing a presentation on myeloma genomics. Thank you, Jonathan.
Thank you. And I guess, can someone confirm you can hear me in the room or online?
Yes.
And you're seeing the slide, everyone's seeing the slides?
Yes.
Yes.
Well, thank you for the opportunity to speak to you. I've been joking with people all day today that it's the first time I get to give a talk on Saturday morning on Friday evening. So the joys of time zones, and I'm as you can see, both Joe McHale and myself are both at TGen and City of Hope. We're actually also the two faculty members here at TGen that are Canadians. And I come from the west coast of the side, and my mother's had the beauty of visiting New Zealand.
Sorry, can I just interrupt? Oh. I think we need to press start slideshow. We're seeing the whole overlay of the slides at the moment. Sorry to interrupt.
Yeah, that's fine. Thank you. I have a feeling it's because you're not seeing the main one.
If you go to the slideshow at the top tab, dual monitor. Yeah, we're seeing your list of slides on the left of the screen and slide 19. Yep. In the center. I'll pass you on to our AV guy, Will.
Okay.
Hey, Jonathan, Will here from AV Media.
Yep.
If you hit escape, oh, hang on, let me just take that slightly down in the room.
Yep.
If you hit escape, so you should just see your slides as normal.
Yep.
Yep. Cool. So if you go up to slide number 1.
Yep.
Yep. So I can see your cursor. Yeah. So if you unshare your screen first, that's probably the easiest way to do it. Cool, yep. So we can just see it. So what I want you to do is I want you to go and start your slideshow as if you're doing like without sharing your screen.
Yep.
Yep. Cool. So you should now be in share screen. And you're on a Mac, is that correct?
That's correct.
Yep. If you hit Command and Tab, it brings up all your different windows, and then just keep hold of Command.
Yep.
Yep. Go over to your Zoom one. Yep. Cool. And then I want you to share your screen. And then when it gives you all the options of which ones you can share, I want you to click on the one that looks like your full presentation, as if it's like the full window. I think it just says like Microsoft PowerPoint presentation. That's the one. Beautiful. Thanks so much.
Perfect.
Thanks, Will.
Thank you, guys. And actually, that was after 2 years of these kind of virtual ones, that was probably one of the best ways of walking through this issue. So thank you very much. Like I was really quickly trying to say, I wish I had been there, because coming from British Columbia, my mother says the only place that's as beautiful as British Columbia is New Zealand. So I'd love to visit someday. So I just wanted to take you through a little bit with the kind of walking assumption that most people here are on the clinical side of myeloma, a little bit of the view of like classic view of myeloma genetics. And then as we go through the little layers, dig a little bit into the nuances that are there that at least I think would be the things I would have a conversation with Joe about, a patient of, well, what about this? So give you some insights there, and then a little bit about understanding the clonal nature of the disease and really trying to bring that back to, as you think about how a patient is performing in front of you over time, what might be going on inside with the tumor mass and the different clones that make up the tumor mass. So just to give you a little bit of kind of where I step back and think about things, really multiple myeloma for the most part, we do know it's a B-cell malignancy. We generally know for the 99% of patients, there's always one or two outliers. They pretty much are all very clearly post-germinal center cells because of the presence of class switch recombination, because of the presence of somatic hypermutation within their BDJ region. We know these are B cells that have gone through that maturation process in the germinal center. And because one of the groups of patients, those that have translocations, which we kind of more scientifically call class switch recombination errors, we know that process is occurring in the germinal center. So we know, quite honestly, that that very first cell that became the cancer cell really did exist in the germinal center of patients. And it eventually migrated out of the germinal center to bring up residence in the bone marrow, we think, as a plasma cell or long-lived plasma cell, and slowly expanded with time to become MGUS clone that was clinically detectable, some additional expansion and maybe additional genetic features, become that diagnostic myeloma clone that required therapy. And then all too often, unfortunately, these patients do progress, and the origin of that progression cell can be varied. Sometimes it's clearly derived from that diagnostic mass of tumors, and sometimes it's from an earlier progenitor. That's some of the work that we've done in the past. And again, just a somewhat easier view of that is this little graphic over here, where we can sometimes view that initial orange circle as that first hit, that very first cell that got something. We know from the work now that myeloma can the origin of that cell can be back 30 plus years in some of these patients. So they sit dormant and slowly grow, slowly accumulating errors. And then that mass that we initially detect, either as a bona fide myeloma diagnosis or as a earlier benign MGUS or smoldering myeloma condition, could be a variety of this part of that tree. And that ultimate part of the tree that leads to progression can be yet another part of that tree, usually having some relationship clearly, but it's not the classic, we keep adding worse things and worse things. It's surprising how often the therapies really do eradicate certain genetic lineages, and what comes back is lacking some things that we thought were clinically important. And some of that, just to highlight, was some of the work we did while I was still at Mayo. I guess, well, both Joe and I were actually at Mayo at the time. This was one of our very first patients that we tracked longitudinally with time, both clinically and then with really comprehensive at the time genetics using comparative genomic hybridization. And we also actually had some very early day whole genome sequencing on this patient through this time point. And what you can see is with time, the patient had a really good response to len with low dose dexamethasone, eventually progressed through carfilzomib, and then did what we've seen all too often, started being exposed to a variety of agents and very quickly blew through every single agent all the way through. We actually started this trying to understand the mechanism of resistance to lenalidomide, because this was a patient who was under continuous lenalidomide exposure, and we wanted to see what was different. Kind of by a little bit by sheer luck, by the time we actually knew about this patient, we'd already had the third sample, and we were able to see that this kind of a pattern that you see on the right, where this is actually chromosome 8, that the 8p region was deleted at diagnosis. When they first progressed after lenalidomide exposure, that 8p deletion had gone away. You'd argue lenalidomide killed off the cells that had it. But this other event, deletion on 8q, appeared. And then when the patient had progressed off of carfilzomib, this is actually some of the early carfilzomib trials, then this patient actually, that 8q deletion had gone away and the 8p came back. And I think Keith Stewart very eloquently called this the clonal tides function by the ebb and flow of the genetics of this patient. What that really did allow us to do in that patient, because having all the technology that we had at Mayo Clinic at the time, we were able to go in and do FISH to really look at these features that we defined by the whole genome analysis, and actually really start to understand what percentage of the cells at each different time point actually made up those different populations that we could track with time. And again, you can kind of see that ebb and flow, but one of the things I want to point out that has changed with today's technology is this group of really dark blue cells, that clone 2.2, there's a little sliver of those cells all the way back at diagnosis. So as things are changing and we start to understand what are the genetic features that really predict poor outcome, we're now starting to get the capabilities to detect those in very small portions of cells, as you can see, and how that will evolve into clinical practice is important. What I think this has really helped people understand more than anything is sometimes when you have a drug that worked really well and a patient progresses, we can think about going back. And I think that's going to be really important as we get into drugs like the BCMA targeted agents. If you give a patient a BCMA targeted CAR T cell and they have a very good response and no progression with no other therapy for 2.5 years, and they then start to progress, are you never going to give them another BCMA agent because you think it's not going to work? And we're going to need to do a lot better job to understand, can you come back with another BCMA targeted agent or not? Because if those agents are so effective and also don't require continuous therapy, making quality of life for our patients better, that's going to be really important to leverage this kind of concept into that population. So let's just back up and talk about what is myeloma genetics. I usually like to tell that it really does break down into two very broad groups when you talk about genetics, and this is going back to the late 70s, 1980s of myeloma, where just using karyotypes, we could break myeloma into two very broad groups of patients, and roughly half the patient population in each of those groups. There's let's talk first, the hyperdiploid group of patients. This is a group of patients that we commonly will refer to as low-risk patients. They generally have trisomies of the odd number of chromosomes. Goal of my life is to understand why it's always the odd number of chromosomes. And we don't understand that yet, but there must be a reason, because if you follow on the AML side, there's actually a similar hyperdiploid group, or actually ALL, and it's the even number of chromosomes. So there's something there that we're just not understanding from a biology standpoint. But then the other group of patients really doesn't have these trisomies. For the most part, they're pretty much normal. We conventionally will call it the non-hyperdiploid, grouping together hypodiploid, which is relatively rare, pseudodiploid, which I would argue is the majority of this group of patients. And then there's a few patients that are near tetraploid, but that's relatively rare, particularly at diagnosis. It increases with time, though. The other group of patients that we have that make up primarily that group of non-hyperdiploid patients are patients that have immunoglobulin translocations. So these are those events that we were very certain for the almost 99% of the time occur in the germinal center, where we break two regions of the genome, one where there's a gene of a potential oncogene, at least in a myeloma tumor cell, and then the immunoglobulin heavy chain enhancer, and we bring those into proximity to lead to the dysregulation. So on the right, you can see a cartoon, a really high resolution cartoon of what happens in a case of 414. There's two thought to be target genes in 414, though I think we've now pretty conclusively said it's this gene MMSET, which we also know as NSD2 or WHSC1. And really what happens in this case is in a normal plasma cell, MMSET is expressed at a constitutive level, but not very highly. FGFR3 is not expressed at all. through the process of the translocation, we bring these enhancers into close proximity with these genes. In the case of MMSET, this intronic enhancer that we call EMU leads to the overexpression of MMSET, taking its expression up quite significantly. And then bringing this really strong 3' enhancer, the alpha enhancer, in proximity to FGFR3 actually turns it on and expresses it as a level similar to what we see for kind of our classic control genes, beta-actin, GAPDH. So it's expressed at an excessively high level in these tumor cells. How do we do this? For the most part today, hopefully most people are no longer ordering cytogenetics, though a lot of places still do. And it's only informative in about 30% of patients. And I would argue is not really changing how you clinically treat a patient, though there's an argument that it helps you understand if a tumor is more proliferative than the average tumor. And I think that is valuable information. There's just other ways to get that measurement. But through FISH, in a normal situation, in the case of this first example with chromosome 13, classic myeloma is looking for either the presence of a 13q14 deletion. This is the locus where the retinoblastoma gene lives, versus cells where both the retinoblastoma and the telomere is missing, really telling us the patient has monosomy 13 loss versus deletion of 13q when you see the loss of the orange and you still have green. So here's an example of one of those patients, normal pattern, red and green, and then we've lost one of those patterns. So one chromosome is lost, or what we would call monosomy 13. The other one to focus on would be this kind of IGH break apart. And we do dual fusion kind of the exact opposite way. In this case, we have two different colored probes on either side of where the breakpoints tend to occur for the immunoglobulin switch translocation. So if you break in between those two colors, you separate those two colors. So we see here in normal, you see a yellowish signal where the two colors are over top of each other. And then here, a very clear situation where there's one yellow and then one green and one red, where those things have totally separated away from each other. As you're looking at these events, the one thing that I would, again, as you're dealing with your local pathology labs who may do this testing, or outside groups that you get the results from, things that we always really recommend to people is really focus on getting what's called the ISCN report. It will help you with interpretations. I deal with a lot of people getting reports that'll say 414 abnormal, but it was really because there was a copy number gain in chromosome 4. There was no T414 event. So sometimes the summaries are not very clear, but if you learn how to interpret the ISCN structure, that's a very standardized nomenclature that has explicit meaning that most groups know how to adhere to. So what events do we think are important, or what are tracked? And I think this is what's been, depending on the site you're at, some of these events are tracked and some of them are not. Most centers will have a part of a panel that will help identify if the patient is hyperdiploid or not. Most often probes targeting chromosomes 5, 9, and 15, but it does vary a little bit by site. There are a few deletions that are frequently tracked, most commonly the 17p or 17p13 deletion that encompasses the TP53 or p53 gene. And then chromosome 13, given its historical significance in myeloma, is still usually used. And then more and more centers are including a 1p31, where, again, as was mentioned by Joe in the earlier talk, 1q events or 1q gain is clearly significant, but there's definitely a relationship between 1q gain and 1p loss. And there's definitely debates about which of those are really the drivers of the poor outcome that we kind of associate with 1q, and I'll show you some of the data on that. More for verbiage, the other translocations that we target, most clinical sites are still doing 414 because it, again, is particularly part of a revised ISS, is one of the high-risk genetic features. The T1416 that dysregulates MAF is also generally done because it's associated with that revised ISS. Unfortunately, revised ISS doesn't include the other MAF translocations that target MAF-B and MAF-A. And actually, if anyone's really paying attention to this slide, that should be 1420, not 1421 for the MAF-B translocation. All those in the data set that we drive with the Multiple Myeloma Research Foundation COMPASS study, all three MAF events have equal prognosis. So those, it's unfortunate that those are not tracked, but they're very rare. They're less than about 2% of all patients. Unfortunately, a lot of centers do not always today, again, being Canadian, maybe biased, do the 1114 anymore because there's never been a outcome associated with it or a prognostic variable associated with it, other than that being generally associated with intermediate or low risk disease. But given some of the data today with venetoclax being more effective in this population of patients, it's becoming more important that it's done because it actually has, for the first time of all of these events, to one of the questions earlier, the potential for a therapeutic recommendation, which is unheard of in the myeloma genetics community today, for all honest truth. There are other translocations that are tracked. The one that's actually much more common than generally appreciated, translocations involving MYC, were largely thought to be progression events, but very clearly these are early events in a lot of patients and is highly associated with hyperdiploid myeloma. So what are the overall frequencies of these? This is data coming from one of our large sequencing initiatives with where I'll just tell you to focus on the WGS column, so the dark green for frequencies. CCND1 is the 1114 target gene, so we track with next generation sequencing any immunoglobulin, so kappa, lambda, or the heavy chain driving the gene. So you can pick up a few events that you'd miss by FISH, but about 20% of patients are positive for 1114. The next most common translocation, again, is the one that we don't generally track, which is the MYC events. What's important there is the MYC translocations are frequently involving the lambda locus, not frequently involving the heavy chain. So even if you do add it to your FISH panel with immunoglobulin heavy chain probes, you'll miss a lot of these events. Most of these events are actually from lambda. And oddly, it's not limited to lambda expressing patients. It's even present in patients who are kappa expressing patients, which was a pretty unexpected finding. Dysregulation of WHSC1, which is MMSET or the 414, again, around 12% of all patients have that event. MAF or 1416 in this case is about 4% in this cohort. And then you can see the other CCND genes and MAF genes. For the most part, they're down in the 1% to 2% range within this cohort. For deletions, again, chromosome 13, and this is probably one of the reasons it lost its favor when we went to interphase FISH from cytogenetics, is about just over 50% of patients have 13q13 deletions. 17p loss is around 10 to 12%. 1q, and I think this is the problem that we have when we struggle with when we talk about being a poor prognostic marker, 35% of patients at diagnosis have a gain, one copy or two copy of the 1q21 locus. And hyperdiploid patients, between 55 and 60% of patients. So still largely half the patients, but a little bit more than half. Now, if we look at from this kind of a sequencing measure and look at copy number, and remember, we just generally break patients into hyperdiploid and non-hyperdiploid. Really what I want to focus your attention on, because I think it changes the way you have a conversation with a patient, is how many different groups of hyperdiploid we can define by hierarchical clustering with this group of patients. And I'm going to point out two groups. So there's the group kind of in the middle that starts with this, what we call HRD classic or HRD ++ 15. Those are really, I'll say, the boring hyperdiploids. We have a group up here that generally don't have chromosome 3 or 7 gains, that just is a biologically interesting subset of patients. And then on the very bottom, we have this group of patients who often do not have trisomies on chromosome 11, but it's been replaced with deletions on chromosome 13 and 1q gains. And the reason I want to point that out is when you start looking at clinical outcome of patients just by these kind of copy number groups, what you actually see, and we're, for time and focus, we'll focus on the progression-free group. If you notice the two groups of patients with the worst progression-free survival, they're actually this group of patients that have hyperdiploid with that 1q gain and deletion 13, and those patients who are not hyperdiploid with 1q gain and chromosome 13 loss. And the reason I want to point that out is as you're talking to a patient who you got a report that says, oh, you have hyperdiploid myeloma, this is good, this is a low risk phenotype. Look at the other markers that you have, because hyperdiploid with no other features, I agree, you can communicate that that's actually a low risk phenotype. But if you have hyperdiploid with 13 loss and 1q gain, that is not necessarily the same group of patients that is that low risk group of patients that we think of when we think hyperdiploid myeloma. Little fun science. Every once in a while, you'll hear that a patient doesn't have a DNA content that is normal. There is a group of myeloma patients that we call near haploid. These are patients that have DNA content of around 0.7, 0.6. So they're not quite haploid, but they're not anywhere close to a typical myeloma patient. If you look at their copy number, what they really have is one copy of all the even number chromosomes and two copies of the odd number chromosomes. This is basically if you looked at a hyperdiploid patient and you subtracted one copy from everyone. These are rare, but it's an interesting thing that lets us start to understand that they have very similar biology clinically, but they also, it's starting to tell us something about the underlying biology of these ratios of these chromosomes. And when you see the learning lesson, watch your assumptions, this is actually one of the patients that I've sequenced the most because our contamination tools here at TGen kept flagging this patient as being contaminated because we never expected this kind of phenomenon. But it also highlights some really unique biology that I think makes us intrigued to what's actually going on in hyperdiploid myeloma. The other thing I wanted to get into from a copy number standpoint is we often talk about 17p deletions by FISH. So we can, using a sequencing-based measure, look at this the same way. So here on the y-axis is the copy number that we see in the tumor, so 2 being normal copy number or 2 FISH signals. Really these dotted lines, anything below this lower dotted line is what we would call high-risk multiple myeloma. Based on a FISH, we would expect this to be positive by a FISH test showing the deletion. When we first plotted this out and overlaid each dot being one patient at diagnosis, and then we colored the dots if they also had a mutation, you really quickly start to notice that all the mutations in P53 are largely in those patients that also have a deletion, leading us to believe that they've lost one copy and mutated the other copy, which is classic tumor suppressor. You have to lose both copies for a tumor suppressor to facilitate the induction of a tumor. Having one copy usually will prevent the tumor. If you look in this same cohort, just doing what we clinically would, looking at that 17p deletion, again, 17p is predictive of outcome, just like we always talk about. It's a very strong clinical predictor. It's why it's in the revised ISS as one of the molecular measures that moves someone into revised ISS stage 3. But if we backed up and just said, what about the biology that's going on here? And let's pretend that we actually read Knutson's 2-hit hypothesis paper. What if we break the patients into those that have normal breaks? They have both copies of P53. Patients where they have lost one copy, but they have the other copy, versus those that are, the car has lost, we cut their lines, the brakes, allelic loss, both copy loss, they have outcomes less, just over 2 years for overall survival. This is in a cohort of patients who are exposed to required to be exposed to a proteasome inhibitor or IMiD or a combination of those. That also does mean that when you're having a conversation with a patient, because they're 17p deleted by FISH, 75% of the time you're having a conversation telling them they have high-risk disease and maybe you're going to change their therapy or maybe you're just going to monitor them more aggressively. 75% of the time, you're wrong. They don't have high-risk disease. And I think when you tell somebody they have low risk and that's wrong, that's not so bad. But when you're telling someone they have high-risk disease, that might change their life decisions. And you really want to have that right. The next question we usually get with that is, well, if you've already lost one copy, aren't you just more likely to lose the second copy? And this is a hard slide to follow, and we have a summary in a second, but I just want to point over here, there are 3 patients that we've tracked with time where they have complete loss, but their complete loss was at diagnosis, and it was complete loss at relapse. Over here, we do have 3 patients who acquired complete loss. In all 3 of these cases over here, these patients did not have a deletion at diagnosis, and they did not have a mutation at diagnosis. They acquired both events, so they were normal. There are some patients where we're acquiring these events, and if you just kind of quickly summarize it, of the limited number of patients that we have, almost 100, but compared to a cohort of 1,000, it's small. About 3.5% of patients transition from being normal at diagnosis to having complete loss of p53 at progression. 10% transition from being normal to partial loss. And then almost 30% of patients, it's small numbers, they go from that partial loss group of patients down to complete loss. So again, if you're trying to talk to a patient, if they have one allele loss, they're 3 times more likely to lose the remaining allele than most patients are to progress and lose 2. They're 10 times more likely to progress to complete loss than a patient with 2 functional alleles at diagnosis. So there's, I think more important there is communicating the risk of moving to this really aggressive phenotype. The other one that we do talk about, and I'll be blunt and say it frustrates me because I do not want to be telling 35% of patients that they have high risk disease, but this is actually a better prognostic variable today in this cohort than 414 is. When you look at 1Q gain, so this is just gain, 1 copy gain or 2 copy gain, it is actually a pretty strong predictor of outcome in this cohort. Though when you start looking at the days, and I can't quickly convert it, but we're talking about predicting worse outcome well out into 5 to 6 years. This is not, I think this is the danger in the myeloma space today. When we see a bifurcated curve like this, and we say the lower curve is high risk, that's a danger. You have to really look at the time, because to me, high risk or ultra high risk is those patients that are not making it through 3 years. A patient who's making it past 5 or 6 years, that's a slightly different term that we should be talking about, because that's also giving them more opportunity to be around to see these newer and newer agents that are coming that are really being transformative. When we do talk about the copies of the tumor, I will just say, because I'm the author on one of the papers that talked about the 2 copy gain being a high risk feature, and it is true. With the analysis that was done there, it's patients who are less than 75 years of age, who are ISS stage 3, and have a 2 copy gain of 1Q versus a 1 copy. The 2 copy do worse. This is a view of all comers, where as of right now in the study, with all comers, not reducing it down to those that have ISS stage 3, not keeping it below age of 75, we don't see a signal in the all comers part. Again, considering the patient you're talking to, if they fit into that revised ISS stage 3, they are under 75 and they have a 2-copy gain on 1Q, I do agree. That's part of that double hit myeloma category. It is a high risk feature that I think you should consider as you think about how to manage that patient. This is kind of the same slide to look at. As an example, this is one of the patients here at TGen that we're following as part of our personalized medicine program. They interestingly had 2 events that we could detect at diagnosis and then when they progressed. One was a whole chromosome gain, and then one was a focal gain of the 1Q region. Things to think when you're thinking therapeutically about 1Q that's worth pulling out. Those patients, the MCL1 gene is generally within the amplified region, especially the small amplified region of 1Q that's commonly used, and there are therapies in trial targeting MCL1. We generally think that's going to be, if it'll ever get working, that is the anti-apoptotic gene that is core to myeloma survival. IL-6 receptor, I think, if like Joe, I'm 20, 25 years into this study, and when I started, everything was focused on expression of IL-6 being a defining feature of myeloma. The IL-6 receptor is on 1Q, so this is something there are, especially with ways that we manage COVID patients, using things that block IL-6 are more common, and maybe we need to revisit them today now that we have better therapies in the myeloma space. And then coming now with FcRL5, this is really the target gene for Servostamab from Genentech, one of the T-cell engager targets that's kind of unique to myeloma, that has a unique play in the space that this is actually expressed on patients that have 1Q, but also expressed at a higher level on patients that have 1Q gain, making it an interesting concept for the future. And in this case, something we flagged for this patient. What we could see with this patient, though, is that clonal evolution idea. Through the genetics we have, and I won't take you through the details, what we could actually tell is that there was 2 groups of cells. One group that had a whole gain of the 1Q arm. Again, when people talk about 1Q gain, what I'll tell you, looking at from a whole genome perspective, 90% of the time it's the entire 1Q gain. It's rarely a focal area of 1Q. But this patient actually did have another population of cells that had a focal gain of 1Q, but having 2 copies. And we could actually track with time between sampling of this patient with time that the gained region, so having 2 extra copies as opposed to 1 extra copy, is winning in this patient. This is actually before any therapy. So this was part of what led us to recommend this patient start therapy. Now, if we talk about translocations in myeloma, I think the ones that we focus on clinically are the 414, so targeting, let me center myself because someone's rotated this, WHSC1 down here in the bottom right-hand corner. And then we also focus on those patients that have MAF here in the upper right-hand corner. What you'll notice here is towards MAF and 414, there's a lot of red lines originating from the immunoglobulin heavy chain locus. But what I do want you to start to appreciate is what we've been missing in this field a lot. There are a lot of translocations originating from the lambda locus. This is stuff we just do not measure today. There's a few coming from kappa. And then there's actually a large diversity of different regions of the genome that target the MYC locus. So we have a lot of events that are underrepresented in the way we look at patients and how we decide if these events are associated with outcome. And that's some of the studies that I'm running, we're working on. We do also have one of the most common translocations that is targeting part of that NF-kappa B process that was mentioned in the introduction. MAP3K14, there's been development of therapeutic agents against this protein. There are hyperactivation of this in a number of patients. And that one's not on here. So we're going to come back to one more later in the talk. So what are we seeing in this cohort? Again, This is all comers. First patient is 27 years of age, oldest patient is 91, but it's a very classic myeloma diagnostic cohort. What we are seeing here is those patients that have those revised ISS features, 414, 1416, in time to second line therapy, which in this observational trial we're using in place of PFS. There's a slight reduction, but no significant difference now, even with the follow-up we have. And in overall survival, there's a little bit of a trend coming at 414, but I think in the post 2010 era, at least again with therapies that are commonly used in the United States, the significance of these events are going to start to come into question as we get into newer and newer agents. I still think they should be discussed, but I don't think, having spent most of my career focused on 414, the therapies are really overcoming the underlying biological difference of that population of patients today. The other one I do want to point out is there are ways to get these same features. So if we just stay on the 414 issue, what we've detected is there are other genes that lead to the dysregulation of WHSC1, or what in the field we'll call MMSET. And today the proper gene name, unfortunately, is NSD2 to add more confusion, but this is the target gene of the 414 translocation. It's usually dysregulated by a translocation, bringing an immunoglobulin enhancer in close proximity, but there are other ways to dysregulate this event. And what you can see is these other genes, some of them are really essential or highly expressed genes in plasma cells, lead to expression that's very similar to we see with those with the translocation compared to those without the translocation. So again, sometimes we're missing patients that have 414 biology by only doing FISH type measurements. We do know there's other ways to look at high risk. So by gene expression profiling, we can see a much higher resolution group of patients. And the only reason I want to point this out is I kind of mentioned that 414 and 1416 maybe are losing their significance. What we can see with gene expression profiling is there's this group of patients that we call PR patients. They have a little bit of all the translocations in them, but this group of patients does very poorly compared to all other patients in this cohort. So we know we can identify these really high-risk patients that, again, have overall survival under 2 years. Considering this is a cohort where median survival is now approaching over 9 years. What we can see with that kind of measure also, and this has meaning when we think about what patients look like at diagnosis versus what they look like at progression, is these patients are acquiring this PR phenotype with time. So more and more patients have this high-risk patient phenotype, and it kind of explains that patient you have that was doing really well, all of a sudden rapidly progressed. You treated them with something you thought was really effective, and they blew through it and succumbed to disease really quickly. They probably went from one of these other groups and dropped into this PR phenotype. We can track what caused that transition. In the case of some of these patients, we know it is that other 1P FISH marker targeting CDKN2C. This is a cell cycle regulator. We think a lot of these patients have lost their control of cell cycle. And as we think about personalized medicine in myeloma and now having BCMA targeted agents, I just want to point out, in this case, this is now looking at almost 1,000 patients, so it's rare. But again, looking 3 ways, we can look at on the y-axis, the copy number of BCMA, the expression of BCMA. Look at this patient up here in the top right-hand corner. It has very high copy number. This is actually going to be 32 copies of BCMA. It has the highest expression of BCMA. We know BCMA is actually overexpressed and drives the NF-kappa B pathway, some of the previous work that I've done. This patient will never be able to get around a BCMA-targeted therapy. That's the patient we could cure today with the agents we have. I would put money in it. This patient down here that has normal copy, but the second highest expression, actually has a translocation, an immunoglobulin translocation dysregulating BCMA. So as we start bringing new technology and these new therapies, we can really start to identify, obviously all these patients we expect to do well with a BCMA targeted agent, but we do have a few patients where they show exceptionally good responses to these agents. Obviously, a lot of this is driven by kind of the changes in the technology, moving away from FISH to sequencing, as we call here at TGen, seq-FISH, because we can make a lot of the exact same measurements from sequencing and give an extra rich layer of data on top of what we would normally look at. And we're now already starting to clinically do this on patients. So patients on the clinical trial that I run, we do this on every patient when they progress, giving the treating oncologists additional therapeutic recommendations based on what was seen. In this case, this patient has a loss of BRCA1, so we can start thinking about a PARP inhibitor that you would never really think about in a myeloma patient. But it does bring up things that have gotten a bad name, I would say, in the myeloma space. So original work that I did with part of the Myeloma Genomics Initiative, we identified BRAF as a common mutation in myeloma. And there's been efforts to look at BRAF and its outcome on patients. And some people show a few good case reports, and generally it's gotten a bad rap, I would argue. But some of this has to do with no one tells you as a clinician frequently what percentage of the cells have a BRAF event. So this is an example of a patient we reported back that they were BRAF positive, right? This was the clinical report for this patient, BRAF V600E positive. When we went and looked at the data after sequencing it, we could actually see that, yes, it's positive, but only about 20% of the sequencing reads showed that. So that means only 40% of the cell mass has it. So actually, this is cellular prevalence, so it was only 20%. So if you completely eradicate 20% of the cells because the BRAF inhibitor works perfectly, would you even detect that clinically? We had a question earlier there about the biomarkers we use today and how they don't respond fast enough. Well, if you eradicate 20% of the tumor mass because the drug worked perfectly, you would never even notice. And the question is, how many good drugs have we thrown away because we weren't really doing the correlative science with those clinical trials correctly? And this can get even more complicated. Yep.
Can I interrupt? Professor Keats, thank you. This is a fantastic talk. I've just been asked to keep to time just so that we can have an ablution break before the next round of speakers. But you're on your last slide. That's fabulous. Thank you.
So sorry to drag on. The big takeaways I wanted you guys to kind of take away from this, particularly, are not all hyperdiploid patients are low risk. So when you're looking at that, think about what other things they have. The deletion of 17p, it is a good predictor of poor outcome. But again, just step back and think, is this the 25% of those patients that have that event that also have lost the other allele and are going to do very poorly? Or are they the 75% of patients who have not? And you're going to give this patient a bad news, but you're not really correct, or there's some of us that would argue you're not correct at a minimum. And then really that there are features beyond what we currently measure, things like the BCMA features that we can measure today that will really predict which patients should get certain therapies as a high priority. Apologize for running a little bit long, and happy to answer questions or over the messages. But thank you very much.
Thank you so much.
Thank you, Jonathan. I know that we are cutting into our break time, but there was a very good question by one of our attendees, which is regarding the TP53. So in New Zealand, what we have decided to do, because of our limited access to treatment, is that based on the EMNO2 data, we say, okay, those who've got deletion 17p who are transplant eligible should be considered for a tandem transplant, just because of the data. So the question is, well, if, as you have presented, that actually 75% of these people aren't actually as high risk as we thought, are we over-treating these patients? And whether, as far as you know, whether the EMNO2 data looks purely just on the presence or absence of 17p deletions, or whether they actually look at whether there was by allelic loss or mutation of the other TP53 gene?
So for the clinical trial design, it was the fish alone. I can't remember seeing something where they've done a subset analysis with the mutational data. Like obviously, I think you are at a risk where you're over treating some of those patients. So maybe they're actually getting. I'd also argue they're getting really effective therapy regardless. So I think that's those hard judgments, right? Like it's a clinically prognostic result, and it's replicated. Everyone replicates that 17p deletion has clinical meaning because it does highlight a population of patients who are going to do bad, but largely because I would argue a subpopulation of those do very poorly, and they just pull the whole group down with them. In the absence of another measure, I think that's a really reasonable thing to do. If you were to then be balancing the cost of a second transplant versus your pathology lab running a cancer panel that they may run for lung cancer that has P53 in it, and should your fish lab be providing cells to them to help you make a decision on balancing that care model, I think you could make an argument that that's not, like, that might actually be a cost savings. And I think that would be the way I would attach that is, whereas for your medical system, I'm Canadian, so I think in a public healthcare system view, where is the actual cost savings, and then also what's best for the patient. And there's a part of me that would argue the tandem transplant is a good thing regardless of the case, but it's hard on the patient, and it does add a large cost in a public system for sure.
Thank you. Are there any questions in the actual conference room? Yes, no?
I can't see any questions here in the floor, so I think we'll break here and come back for 11 o'clock. Does that sound all right, Henry?
Yes, sounds perfect. And thank you, Jonathan. Thank you for your time. Great talk.
Yeah, thank you.
Yeah.